This lecture focuses on the cellular level of organization, which is where structure and function in the human body truly begins. Cells are the smallest living unit, but they're also the foundation for everything we study in anatomy and physiology. By understanding how cells are built, how they function, and how they divide and specialize, we can better understand how tissues, organs, and organ systems emerge. Throughout this lecture, we will connect cellular structure to cellular function and see how disruptions at the cellular level can ultimately affect the entire organism. This perspective will guide how we approach the rest of this course. In this lecture, we are building a clear roadmap for what we should be able to do by the end of this unit. You should be able to describe the structure and function of the cell membrane and explain how it regulates what enters and leaves the cell. You should also describe the function of the major cytoplasmic organelles and explain how the nucleus is structured and what it contains. In addition, you should be able to list the stages of the cell cycle in correct order, including the specific steps involved in somatic cell division. Finally, we will discuss how cells differentiate and become specialized, even though they all contain the same DNA. These objectives reflect the core skills and concepts that connect cell structure to cell function and development. The cell is defined as the smallest structural and functional unit of life, meaning it is the simplest level at which life exists. All living organisms, whether simple or complex, are made up of one or more cells. This idea comes from cell theory, which tells us that cells are the basic unit and structure and function in all organisms, and that new cells arise only from pre-existing cells through division. In the human body, we recognize two main categories of cells: somatic cells, which make up tissues and organs, while sex cells or gametes include the egg and the sperm cells and are also involved in reproduction. In anatomy and physiology, understanding cells is essential because every tissue, organ, and body system depends on cellular processes like metabolism, communication, and cell division to function properly. Every human life begins as a single fertilized egg, yet that one cell contains all of the genetic information needed to build the entire organism. The repeated rounds of mitosis, that original cell divides again and again, producing trillions of cells. Early in development, these cells are undifferentiated, meaning they have not yet taken any specific roles. Over time, signals within the body guide cells through differentiation, allowing them to become specialized. This progression from one cell to many cells highlights how cellular processes drive growth, development, and organization. Although all cells contain the same DNA, they do not all look or behave the same. Cell specialization allows structures to match function. For example, flat, tightly packed skin cells form a protective barrier, while neurons develop long extensions that allow them to transmit electrical signals across long distances. These differences arise from gene expressions, where certain genes are turned on or off depending on the cell's role. This specialization increases efficiency and allows tissues and organs to perform highly specialized tasks essential for survival. Despite the incredible diversity of cells in the human body, they share a common organizational plan. Most cells are built around three main components: a boundary that separates the cell from its surroundings, an internal fluid filled region where cellular activity occurs, and a control center that houses genetic materials. Within the cell, specialized structures called organelles carry out specific tasks, which, like departments within a factory. Variations in size, shape, and internal organization allow different cells to perform very different functions while following the same basic blueprint. One of the most important ideas in biology is that the structure and function are inseparable. Biological structures are shaped by the job they perform, from the microscopic level of cells to the macroscopic level of organs. When a structure is well matched to its function, the result is efficiency and stability, which supports homeostasis. A classic example is the red blood cell, whose shape enhances oxygen transport, or intestinal cells, whose surface features increase absorption. Every living cell is enclosed in a cell membrane that defines its boundaries and protects its internal environment. This membrane acts as a gatekeeper, carefully controlling what enters and leaves a cell. By regulating the movement of nutrients, waste, and signaling molecules, the membrane helps maintain internal balance. At the same time, it allows the cell to interact with its surroundings. Without this selective barrier, cells would not be able to survive or function properly. The cell membrane is not a rigid wall but a dynamic and flexible structure. Its foundation is a phospholipid bilayer arranged so that the water-loving regions face outward and the water-fearing regions face inward. Embedded within this layer are cholesterol molecules that help maintain fluidity and proteins that carry out transport, signaling, and structural roles. The phospholipid bilayers also have a unique dual nature that makes membranes formation possible. Each molecule has a hydrophilic head that interacts easily with the water and hydrophobic tails that avoid water. When placed in a watery environment, these molecules naturally arrange themselves into a bilayer. This arrangement creates a semi-permeable barrier that blocks some substances while allowing others to pass. Proteins embedded in the cell membrane carry out many of the membrane's most important functions. Some proteins span the entire membrane and act as channels or carriers that move substances into or out of the cell. Others serve as receptors, allowing the cell to receive and respond to chemical signals. Proteins attached to the membrane surface can function as enzymes, anchors, or recognition markers. Together, these proteins give the membrane its functional specificity and allow the cell to interact precisely with its environment. Extending from the outer surface of the cell membrane are carbohydrate chains attached to proteins and lipids. These structures collectively form a glycocalyx, which plays a critical role in cell identity and communication. The glycocalyx allows cells to recognize one another, distinguish self from foreign cells, and adhere to neighboring cells. It also participates in cell signaling and provides a layer of protection for the cell surface, especially in tissues exposed to friction or harsh environments. Some cells possess specialized surface structures that enhance their function. Microvilli are small projections that increase surface area and are especially important in absorption in the digestive tract. Cilia are short hair-like structures that move fluid and particles across the cell surface, such as mucus in the respiratory system. Flagella are longer structures that enable cell movement, most notably seen in sperm cells. These surface adaptations reflect the specific functional demands placed on different cell types. The cell membrane is selectively permeable, meaning it allows some substances to cross while restricting others. This selectivity is essential for maintaining a stable internal environment. Transport across the membrane occurs through two major categories: passive transport, which does not require energy, and active transport, which requires cellular energy in the form of ATP. By carefully regulating a movement across the membrane, the cell controls their internal composition and responds to changes in their surroundings. Passive transport moves substances down their concentration gradient from areas of higher concentration to lower concentration without the use of ATP. This process includes simple diffusion, facilitated diffusion through membrane proteins, and osmosis, which specifically moves the movement of water. Passive transport is driven by natural molecular motion and plays a major role in maintaining equilibrium between the cell and its environment. Active transport allows cells to move substances in directions that passive transport cannot. This process requires energy, usually in the form of ATP, because materials are moved against their concentration gradient, from areas of lower concentration to higher concentration. Specialized membrane proteins act as pumps or carriers to accomplish this movement. Active transport is essential for maintaining ion balances, nutrient uptake, and overall cellular function. Endocytosis is a form of active transport that allows cells to take in materials that are too large to pass directly through the membrane. During this process, a portion of the cell's membrane surrounds the materials and pinches off to form a vesicle inside the cell. Phagocytosis involves engulfing large particles such as bacteria, while pinocytosis brings in fluids and dissolves substances. Receptor-mediated endocytosis allows cells to selectively take in specific molecules based on receptor binding. Cells rely on multiple active transport mechanisms to maintain proper internal conditions. Carrier-mediated transport uses specific proteins to move substances across the membrane using energy. Vesicular transport, including endocytosis and exocytosis, move materials in bulk using membrane-bound vesicles. These mechanisms ensure that essential molecules enter the cell while waste products and signaling molecules are properly managed. Carrier-mediated transport involves proteins that bind specific substances and undergo a shape change to move them across the membrane. This process is highly selective and energy dependent. Vesicular transport, in contrast, packages materials inside membrane-bound sacs for transport in or out of the cell. Together, these processes allow cells to handle both small molecules and large particles efficiently. Osmosis refers to the movement of water across a selectively permeable membrane, often through specialized channels called aquaporins. Tonicity describes how surrounding solutions affects cell volume. In an isotonic solution, water movement is balanced and cell size remains stable. In hypotonic solution, water enters the cell, causing the cell to swell, while in hypertonic solutions, water leaves the cell, leading to shrinkage. These principles are especially important for understanding fluid balance in the body. Exocytosis is an active transport process used to move materials out of the cell. In this process, vesicles inside the cell travel into the cell membrane, fuse with it, and release their contents into the extracellular space. This mechanism is essential for secretions including releasing hormones, neurotransmitters, and digestive enzymes. Exocytosis also plays a role in immune response by allowing cells to release signaling molecules that coordinate defense and inflammation. The cell's membrane serves as both a protective barrier and a regulatory interface between the cell and its environment. Its structure allows flexibility while maintaining control over what enters and exits the cell. Through membrane-bound proteins, carbohydrates, and transport mechanisms, the membrane supports communication, nutrient exchange, waste removal, and internal balance. Inside the cell is the cytoplasm, which is a region where most cellular activity takes place. The cytoplasm includes the cytosol, which is a fluid rich in enzymes, ions, and nutrients, as well as a variety of organelles with specialized roles. In eukaryotic cells, these organelles are often membrane bound, allowing different processes to occur simultaneously without interference. Proper coordination within the cytoplasm is essential for normal cell function and survival. The endomembrane system is a group of interconnected organelles that work together to synthesize, modify, transport, and degrade cellular materials. Key components include the endoplasmic reticulum, Golgi apparatus, vesicles, and lysosomes. Proteins and lipids are produced, processed, and directed to their appropriate destination through this system. Efficient coordination within the endomembrane system allows cells to maintain organization and meet changing functional demands. The endoplasmic reticulum, or ER, is divided into the rough and smooth regions based on structure and function. Rough ER is studded with ribosomes and is involved in protein synthesis and initial modification. Smooth ER lacks ribosomes and plays a role in lipid synthesis, detoxification of harmful substances, and calcium storage. Together, these regions support a wide range of metabolic and regulatory activities necessary for cell maintenance. Proteins and lipids produced in the endoplasmic reticulum are sent to the Golgi apparatus for further processing. Here, molecules are modified, sorted, and tagged as they reach the correct destination. The Golgi packages these materials into vesicles that either move into or other organelles or travel to the cell membrane for secretion. This step ensures that cellular products are delivered accurately and efficiently. Lysosomes are membrane-bound vesicles filled with powerful digestive enzymes. Their primary role is to break down worn-out organelles, cellular debris, and foreign materials such as bacteria. This recycling process, known as autophagy, helps maintain the cells healthy by removing damaged components. Lysosomes also play an important role in immune defense and in programmed cell death when cells are no longer functioning properly. Mitochondria are responsible for producing ATP, the main energy currency of the cell. They have a double membrane with the inner membrane folded into structures called cristae that increase surface area for energy producing reactions. Cells within high energy demands, such as muscles and nerve cells, contain large numbers of mitochondria. Proper mitochondrial function is essential for supporting metabolism, movement, and cellular activity. Peroxisomes are small membrane-bound organelles that contain enzymes involved in detoxification and lipid metabolism. They break down fatty acids and neutralize toxic substances, including harmful byproducts of cellular metabolism. Peroxisomes also produce and degrade hydrogen peroxide, preventing damage from reactive oxygen species. These functions are especially important in cells in liver and the kidneys. The cytoskeleton is a dynamic network of protein fibers that extends through the cytoplasm. It provides a structural support, maintains cell shape, and allows cells to move and change shape when needed. Components of the cytoskeleton also guide the movement of organelles and vesicles and play a key role in cell division. The nucleus is typically the largest organelle in the cell and serves as the primary control center. It is enclosed by a double membrane called a nuclear envelope, which contains pores that regulate and enter the exit the cell. Inside the nucleus houses the cell's genetic material and coordinates activities such as growth, metabolism, and division. While most cells contain a nucleus, there are notable exceptions, including red blood cells and some cells like skeletal cells that contain multiple nuclei. Within the nucleus, DNA is organized into a form known as a chromatin, which consists of DNA wrapped around histone proteins. This arrangement allows long strands of DNA to fit inside the nucleus while remaining accessible. The nucleolus is a dense region where ribosomal DNA is produced and ribosome subunits begin assembly. Surrounding these structures is the nucleoplasm, a fluid environment containing enzymes and nucleotides needed for nuclear processes. The nucleus stores and protects the cell's DNA, ensuring genetic information remains intact. It directs protein synthesis by sending messenger RNA instructions to the cytoplasm. The nucleus also regulates cell growth, repair, and division by controlling gene expression and coordinating DNA replication. These regulatory roles are essential for maintaining normal cell function and preventing errors during cell division. DNA is composed of two strands forming a double helix built from repeating units called nucleotides. Each nucleotide contains a sugar, a phosphate group, and a nitrogenous base. Bases pair specifically with adenine pairing with thymine and cytosine pairing with guanine. Segments of DNA known as genes carry instructions for protein production. Depending on the stage of the cell cycle, DNA exists as loosely coiled chromatin or as tightly condensed chromosomes. Transcription is the process by which the information stored in the DNA is copied into messenger RNA. This occurs in the nucleus, where the DNA strands separate and an enzyme called RNA polymerase builds a complementary RNA strand. Once formed, the mRNA detaches and exits the nucleus through a nuclear pore. This mRNA then carries genetic instructions to the ribosome in the cytoplasm, where proteins will be assembled. Once messenger RNA reaches the cytoplasm, translation begins at the ribosomes. During this process, the ribosome reads the mRNA sequence in groups of three bases called codons. Transfer RNA molecules bring specific amino acids to match each codon, allowing them to be linked together in a correct order. Ribosomal RNA forms the structural core of the ribosome and helps catalyze this assembly. The end result is a protein whose structure and function is determined by its original DNA sequence. Most cells in the body divide to support growth, development, and maintenance. Cell division allows damaged or worn out cells to be replaced and enables tissues to heal after injury. Not all cells divide regularly, however. Red blood cells lack nuclei and cannot divide, while many neurons and muscle cells divide very rarely or not at all. Understanding which cells divide and which do not helps maintain patterns of regeneration and repair in different tissues. The cell cycle describes the sequence of events a cell goes through from one division to the next. It consists of two major phases, interphase and the mitotic phase. Interphase is the longest portion and includes periods of growth, DNA replication, and preparation for division. The mitotic phase includes mitosis and cytokinesis, resulting in the formation of two new cells. This cell cycle ensures continuity of life at the cellular level. During most of the cell's life, DNA exists as loosely packed chromatin, which allows genes to be accessed and expressed. During the S phase of interphase, DNA is replicated, producing identical copies known as sister chromatids. As the cell prepares to divide, chromatin condenses into highly packed chromosomes. Sister chromatids remain joined at the region known as the centromere until they are separated during mitosis. Mitosis occurs in four distinct stages that ensures accurate distribution of genetic materials. During prophase, chromatin condensed at the mitotic spindle begins to form. In metaphase, chromosomes align at the center of the cell. Anaphase follows as sister chromatids are pulled apart to opposite poles. During telophase, nuclear envelopes form and chromosomes begin to condense. setting the stage for cell division to be completed. Cytokinesis is the physical division of the cell that follows mitosis. During this process, the cytoplasm and organelles are separated to form two distinct daughter cells. In animal cells, this occurs through the formation of cleavage furrows that pinches into the cell into two. After mitosis divides the nucleus, cytokinesis is what truly completes cell division and allows the two new cells to function independently. Cell division is tightly regulated to ensure accuracy and prevent damage. The cell cycle includes checkpoints that monitor cell size, DNA integrity, and chromosome alignment. At the G1 checkpoint, the cell assesses whether conditions are favorable for division. The G2 checkpoint verifies that DNA replication is complete, while the spindle checkpoints ensures chromosomes are properly aligned before separation. These control mechanisms protect the organism from mutations and uncontrolled cell growth. Progress through the cell cycle is controlled by interactions between cyclins and cyclin-dependent kinases, or CDKs. Cyclins are regulatory proteins whose levels rise and fall throughout the cycle, while CDKs are enzymes that become active when bound to cyclins. Together, cyclin-CDK complexes trigger transitions such as entry into the DNA synthesis or mitosis. This precise regulation ensures that each phase of the cell cycle occurs in the correct order. When the cell cycle regulation fails, cells may divide uncontrollably, leading to cancer. This loss of control often results from mutations in genes that regulate checkpoints, DNA repair, or program cell death. Tumors can be benign, remaining localized, or malignant, invading surrounding tissues and spread into distant sites. The immune system plays a critical role in identifying and eliminating abnormal cells before they become a serious threat. Cell differentiation is the process by which unspecialized cells develop into specialized cell types. Although all cells contain the same DNA, differences arise because specific genes are turned on or off. Transcription factors help control gene expression, guiding cells towards particular structures and functions. This process allows a single fertilized egg to give rise to the wide variety of cells that make up the human body. Stem cells are classified based on their developmental potential or potency. Totipotent cells, such as the zygote, can give rise to all cell types in the body as well as extraembryonic tissues. Pluripotent stem cells can differentiate into any body cell but not placental tissues. As development progresses, stem cells become more restricted. Multipotent stem cells produce a limited range of related cell types, such as blood cells. Oligopotent and unipotent stem cells are even more specialized, contributing to tissue maintenance and repair throughout life. Stem cells have significant potential in medicine because of their ability to self-renew and differentiate. They are used clinically in treatments such as bone marrow transplants to replace damaged or diseased blood cells. Researchers are exploring stem cell therapies for conditions including neurodegenerative diseases, heart damage, and spinal cord injuries. In addition to therapy, stem cells are valuable tools for studying development, modeling diseases, and testing new drugs. These applications highlight how understanding cellular biology translates directly into advances in healthcare.